Hello friends, good evening. Today I am going to solve GATE NAT questions of year 2024 biochemistry paper. In the previous video, I have solved the two questions from the same biochemistry paper of year 2024. Today I am going to solve next two questions from the same year and same paper. Let's solve. GATE question 35. If a 10 millimolar solution of a biomolecule in a cuvette of path length 10 millimeter absorbs 90% of incident light at 280 nanometer, the molar extinction coefficient of the biomolecule at this wavelength is. Expected answer should be in the range of 98 to 102, and answer should round off to 2 decimal places. In this figure, this is a cuvette, and the first line is the incident light that passes through this solution in this cuvette, and the second line is a transmitted light. Incident light is denoted as I naught and transmitted light is denoted as I. Incident light is 100%, and in this question the solution is absorbs 90% of incident light. That is, in this cuvette there is a solution and it absorbs 90% of light, and so transmitted light would be 100 minus 90, that is 10%. So this is incident light and this is incident light and this is transmitted light. So by using this concept we can calculate the transmittance. To solve this question, we should first use three formulas to solve. That is, first is a Beer Lambert's law formula, that is absorbance is equals to molar extinction coefficient multiplied by concentration multiplied by optical path length. Okay, this is the Beer Lambert's law equation. And second equation is a transmittance, that is transmittance is equals to transmitted light divided by incident light. Okay, and the third is absorbance is equals to log of 1 upon transmittance. Okay, now to solve this Beer Lambert's law equation, we have concentration, we have optical path length, but we have to find absorbance to calculate the molar extinction coefficient. So to find the absorbance we have to find the transmittance, because in this equation, in this formula, by using the transmittance we can find the absorbance. So transmittance, let's find the transmittance. Before going to find transmittance, I wanted to tell you that here the solution is given in millimolar, okay, and path length is given in 10 millimeter. We have to convert. To solve this Beer Lambert's law equation, concentration should be in the molar unit and path length, optical path length, should be in the centimeter unit. So let's convert first. That is 10 millimolar, you have to convert to molar. So millimolar, to convert in millimolar to molar, you have to divide by 1000. So 0 get cancelled, that is 0.01 molar. Concentration is 0.01 molar. Simultaneously we have to solve for path length. Path length is 10 millimeter. You have to convert into centimeter. So 10 divided by 10. To convert to centimeter from millimeter, you have to divide by 10. So you will get 1 centimeter. Okay, I'll write down in some other area that the concentration is equals to 0.01 molar and optical path length is equals to 1 centimeter. I'll make some space to solve further. Now we have to solve the you have to find the transmittance first, and then after finding the transmittance we can find the absorbance. Okay, so Transmittance is equals to transmitted light divided by incident light. So transmitted light is what? Transmitted light is this light. So it is 10 divided by incident light. This is the incident light, that is 100. You cancel, that is 0.1. Transmittance is equals to 0.1. By using transmittance is equals to 0.1, we can solve absorbance is equals to log 1 upon 0.1. So 1 upon 0.1 is A is equals to log 1 upon 0.1 is log 10. So absorbance is equals to 1. Log 10 is 1. So I'll write down here absorbance is equals to 1. Now we have this all concentration, optical path length and absorbance. Now we can use this all units to find the molar extinction coefficient. So here is a formula. Keeping the molar extinction coefficient in the right hand side, shifting concentration and optical path length to the left hand side, get like so. Substituting all the values, 1 upon concentration is 0.01 multiplied by optical path length is 1 is equals to molar extinction coefficient. By solving this, 1 upon 0.01 multiplied by 1, you will get the 100 point. Molar inverse, centimeter inverse. This is a unit for molar extinction coefficient. So this is the final answer. In my calculator, I got this answer directly. After decimal point, there are no digits, so I have stopped the calculation here. So 100 is my answer. The molar extinction coefficient of biomolecule at this wavelength is 100 molar inverse centimeter inverse. Let's go to next question. GATE question 44. In Michaelis-Menten's equation, if substrate concentration is equals to 15 km, then the ratio V0 upon Vmax. Expected answer should be in the range of 0.93 to 0.95, and answer should be round off to 3 decimal places. V0 upon Vmax. V0 is initial velocity of the reaction and Vmax is the maximum reaction velocity achieved at saturation. Now what do you mean by saturation? Saturation is a point where all enzymes active sites are occupied by the substrate. So we have to solve this ratio, we have to find this ratio. First I'll write the Michaelis-Menten equation, that is V0 is equals to Vmax multiplied by substrate concentration divided by Km plus substrate concentration. Okay. So V0 is equals to Vmax, substituting substrate concentration, that is multiplied by 15 km divided by Km plus 15 km. Okay, so V0 is equals to Vmax multiplied by 15 km divided by Km, 1 km plus 15, that is 16 km. So taking this Vmax to the left hand side in the division, that is the ratio they are asking is V0 upon Vmax. So taking Vmax to the left hand side in the division, Vmax, V0 upon Vmax is equals to 15 km divided by 16 km. Km get cancelled, so V0 upon Vmax is equals to 0.9375. Now this is not our final answer because we have to round off to 3 decimal places. So after point 1, 2, 3, third digit, third digit, if it is greater than 5, you have to add +1 to this digit and then solve. Then it is rounded up to 3 decimal places. So that is V naught upon Vmax is equals to 0.937 plus 1 is 8. So this is the final answer. You need not worry about the round off to 3 decimal places or round off to 2 decimal places, because in the answer key they have rounded off to 2 decimal place. Okay, so if it is coming in your range, even if you write like this, your answer will be absolutely correct. So no need to worry. Okay, thank you. This is the end of the video. See you in the next video.